Hi there, my name is Dylan. I'm a PhD student in the Movement and Exercise Neuroscience Lab at Monash University. And here I'll be presenting work done with my supervisor, James Coxon, and a former honours student in our lab, Melissa Pally. And we were looking at the combined effects of two types of non-invasive brain stimulation. So intermittent theta burst stimulation, ITBS, is one type of non-invasive brain stimulation that can modulate long-term potentiation like plasticity within the cortex. And while the technique has important research and clinical applications, one downside is that its after effects can be variable across individuals, hindering translations to these settings. Combining ITBS with other types of non-invasive brain stimulation, such as transcranial alternating current stimulation, or TACS, may be one way to produce more consistent effects, although this hypothesis remains preliminary. So in this study, we explored whether applying gamma frequency TACS before ITBS could enhance its after effects. And to do this, we used a repeated measures double-blind design. So as shown in the main figure, all participants completed 2 sessions. One session they received a gamma frequency TACS at 75 Hertz over the hand area of the primary motor cortex. And in another session, they received the sham control. And to quantify these effects, we used 3 TMS measures of excitation and inhibition. And they were corticomotor excitability, short interval intracortical inhibition, SICI, and intracortical facilitation, ICF. And so each of these 3 measures were collected at baseline, again after TACS, and then again after theta burst. So onto our results. And here I'm showing 3 graphs related to each of our 3 TMS measures. And just to briefly orient you, on the x-axis of each graph is the various time points, and on the y-axis is the size of the mean response, or for SICI and ICF, the ratio response. And in the 2 lines in each graph show the conditions. So gamma TACS is shown in the gray and the sham is shown in black. And as can be seen in the graphs, and as indicated through our statistical analyses, we observed no significant main or interaction effects. So overall, we found that applying gamma frequency TACS before ITBS did not enhance its after effects. And one possible reason for this could be related to metaplastic mechanisms, particularly given the extended length of our TACS protocol, so 20 minutes versus 3 minutes used in another previous similar study, although this would of course need to be verified in future research. Thank you for listening. I'd be happy to answer any questions or chat about any of the results if you're interested.